Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at Module 5, 5.1.2, excretion as an example of homeostatic control. And we're going to start with the kidney structure. So you should know that you have got two kidneys inside your body. Now the kidney's role in this homeostatic processes, the control within our body, is one, that it's going to filter the nitrogenous waste products. So linking back to your liver topic when we looked at deamination, we're going to remove the amine group from our excess amino acids. That's going to result in ammonia. That ammonia then gets converted into urea through the process of the ornithine cycle. So what we need to do is we need to filter this nitrogenous waste, this urea that has been produced at the liver. So therefore it needs to be transported from the liver to the kidneys, and it gets transported in the blood through that renal artery. Well, the kidney's role is not only about the excretion, this filtering of the nitrogenous waste, it's also about osmoregulation, about maintaining the water balance and the pH of the blood, therefore maintaining that of all the tissue fluid surrounding the body. Therefore maintaining the water balance surrounding the cells. So it's important that we note that the blood is being supplied to the kidneys via the renal artery, and the filtered blood that comes out will be taken back into circulation through what's known as the renal vein. So all of the substances that are going to be excreted will actually move from the kidneys down through the ureter into the bladder, where it's stored until we are ready to excrete it from the body, and we excrete it through the urethra. So it's important that we note the kidney's role in the homeostatic mechanisms of excreting the nitrogenous waste, which would be toxic, and also maintaining that balance in our water and pH, so that osmoregulation. So let's actually look into the kidney. Let's look at how we would actually dissect it. Now, when you go to dissect a kidney, it is very soft and it is very difficult to do that dissection without destroying too much. So you have to take real care with the dissection of a kidney. So this is what it looks like on the outside, and when you get the opportunity to open and dissect it, what you're actually going to identify is that there's three main areas. So the three main areas that you need to understand and note is the first part, which is the cortex. Now the cortex is a darker color, and the reason it's a darker color is because this is the area where the majority of the blood vessels are leading to. We then move down into our medulla, and in our medulla is where our kidney nephrons actually flow down into the pelvis area, which is the last collecting basin, which is collecting all the urine that's going to be taken down to the ureter. So what will they expect you to do in exam? If they give you a question which is showing you the structure of a kidney, they are more than likely going to test your biological drawing skills. So whenever you do a biological drawing, it's really important that you remember these key notes. You must use a really sharp pencil. You must fill at least half the space that they've given you in the exam. You need to make sure that it's correct proportions. Now, it's really important that whatever you are drawing is a likeness to the picture that they have shown you. So don't try and draw a textbook diagram. Make sure that what you are drawing is a likeness of what you could see. You should use a ruler to label your lines, and do not let those label lines cross. Also, they must touch the part that you are labeling, and it must be clear. So it's really important whenever you do your diagram that you make sure that you haven't made any parts which are missing or squiggly lines. If you do make mistakes, you should use a rubber to rub it out and then redraw it. So make sure that you do have your labels, which are obviously your cortex, your medulla, and your pelvis. So when they are looking for a biological drawing, they are the three parts that they want to see drawn and labeled, and you want to make it look as close to the picture that you've been given. Now, they're not expecting you to be an artist, so therefore they want to see it looking vaguely like what you have in the picture. So don't stress out that it has to look amazing, because it doesn't. So if we actually looked at the kidney nephron, then there are some key parts that you need to know. Now, as I said before, the cortex, the darker section, has the majority of the parts of the kidney nephron. So let's just look at that now. So the first thing you have to know is what's called the glomerulus. So the glomerulus is the bundle of capillaries. So it's a capillary bed which sits inside a Bowman's capsule. Now this is where ultrafiltration is going to occur, and I will do a video on ultrafiltration, so do check that one out. We then move through the kidney nephron from the Bowman's capsule to the proximal convoluted tubule. Now you will hear this referred to as the PCT. Make sure in the exam that you have at least written it once as the proximal convoluted tubule before you start abbreviating. We then go down through the loop of Henle. It's got a descending limb and an ascending limb, and then we lead on to our distal convoluted tubule. So as you can see from my diagram so far, the only part which actually falls down into the medulla section is the loop of Henle at this point. Everything else, the glomerulus, the Bowman's capsule, the proximal convoluted tubule, and the distal convoluted tubule, all sit within the cortex. Remember the darker, redder area of the kidney, and that is where majority of this will be found. If we go down into the medulla, we are looking at the loop of Henle and the collecting duct. So these are the only parts which are found within the medulla, and then that medulla, the collecting duct, will then lead to the pelvis, where the urine will collect before it moves down through the ureter. Well, I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, do check out my revision platform, www.aiqchat.com.